Good morning, good evening to everyone joining with us today from around the world to the 13th episode of PhD Talks by NcodeX. So we are gathered here for the 13th episode of PhD Talks by NcodeX with Dr. Dilshan Gunasinghe here. He is currently a postdoctoral research fellow at University of New South Wales and Monash University. Welcome, Dilshan. We are happy to have you.
Thank you for having me.
Before we move on, let me give you a brief introduction to who we are, a little bit introduction to NcodeX. So NcodeX's vision is to become the pioneer in interdisciplinary STEM, science, technology, engineering, and mathematics R&D innovation for a sustainable future by bridging the long prevailing gap between academia and industry. So how do we do that? By innovative research development and execution, and also STEM coaching and training for young researchers to meet the global demand. So we gather early career researchers, senior scientists, professionals from in either academia or industry, and bring them into one roundtable to brainstorm ideas, share knowledge and experiences and expertise to collectively produce innovative, trending, global scale research and provide a sustainable, innovative solutions to meet the global challenges. So to know more about NcodeX research and our available training programs, please visit www.ncodexpert.com, which is also given in the chat box or in the comments. Right, so today on the 13th episode, Dilshan will be talking about demystifying cellular biology with nanoscopy technology. So for any questions and discussion points, please post them in the chat box or in the comments, and we will discuss them after the presentation during the Q&A session. So without further ado, Dilshan, over to you.
Thanks, Rory. Thanks for the introduction and thanks for the opportunity. So I'll share my screen now. Okay, hopefully you guys can see my screen. Hello everyone, I'm Dilshan. I'm a research fellow in a single molecule imaging laboratory in University of New South Wales, Sydney, and I'm also a visiting research fellow at the moment at Monash University. So my talk today is about demystifying cellular biology with nanoscopic technology. Now, I understand this maybe perhaps be a bit of a broad title, but I will surely try to concise my research and talk in a more general term, so to reach a more general audience in a way. And so, again, my focus is going to be on this nanoscopic technology, which is leaps ahead on microscopy, which we all know. In STEM field, everyone had some form of taste of microscopy in their childhood or in their school years. And so my contribution to this technology has been coming from bacteriology and particularly T-cell biology. Now, before we go into the topic of nanoscopy, I would like to talk to you about the fluorescent microscopy and how it's intertwined with cell biology itself. Now, fluorescent microscopy had played a very crucial central role in describing biology itself, especially molecular organization and interactions in biological systems. Its high molecular specificity and its imaging capabilities span beyond single color imaging, which we all know with light microscopy had, which sort of expands with fluorescent microscopy. And this is also a very direct visualization method used in biology to study specific molecular species and their interactions in a very non-invasive manner, which ultimately provide means to study living systems under physiological conditions, which is what biologists really need. However, one of the main challenges in fluorescent microscopy comes from its diffraction limited spatial resolution set by the diffraction limit of light, or the diffraction limit itself, in simple terms. And this is described by Ernest Arbey in 1873, which is coined as the Arbey's diffraction limit, which shows the highest resolvable object size from a light microscope was to be 0.25 microns. So with the advent of super resolution microscopy or nanoscopy, these terms are interchangeably used in the field. So I will be referring to nanoscopy as super resolution microscopy and vice versa, but it means the same thing. And so with the advent of these type of high resolution microscopy techniques that circumvent this limit of diffraction limit, opens up new avenues to directly visualize macromolecules in biological systems. So if you look into the brief history of microscopy, you can see it basically revolutionized the field of biology, in particular cell biology itself, coining the term cell, as it seems, in 1665, to the invention of electron microscopes, and then getting the Nobel Prize in 1986, and then of course in 2014, again for the invention of nanoscopy itself. In this presentation, I will be using a lot of these technical terms, but I will try to make it more general so that all the audience can understand. So what are these nanoscopic techniques? There are three main nanoscopic techniques that we know of, being the very inception of these techniques. The pioneers of these techniques actually got the Nobel Prize in 2014 for chemistry, actually, but the applications are almost always for biology. Namely, structured illumination microscopy, stimulated emission depletion microscopy, and single molecule localization microscopy. Now, structured illumination microscopy, in short for SIM, uses Moore pattern, which is a pattern illumination technique of fluorophores, and use this Fourier transformation mathematical technique to create a twofold improvement in the resolution limit. Now, STED microscopy reduces the size of the diffraction limit, the spot by using point spread function engineered to achieve 5 to 10-fold increase in the resolution. Now, point spread function, or PSF, is basically can be described as a measure of blurriness induced by the diffraction of light due to the experimental setup, and also expresses as a normalized intensity distribution of a point source, which in this case, for a lot of these fluorescent microscopy techniques, is a fluorophore. And my focus will be on single molecule localization microscopy technique, which I've been using for 5 years, more than 5 years now, to be honest, which by far offers the highest resolution of these techniques, allowing quantitative and statistical capabilities. It relies on this method of temporary resolving these images that we take, and then finally reconstructing this final resolved image up to about 30 nanometer scale resolution, using this, again, technique called Gaussian fitting, which is based on pointillism, which I won't be going into more detail on this. But yeah, the final reconstructed image would actually have a resolution of around 25 to 30 nanometers, which is very close to the scale of macromolecules in biological systems. So that's a very big breakthrough in these nanoscopy techniques. So nanoscopy in general have been branched into numerous subfields in biology, and it continues to expand, and it's ever evolving. And my contributions are primarily lies in bacteriology, which comes under host pathogen interactions, and also T-cell biology, which comes under immunology. And so recently, with more inventions of these techniques, there even greater super precision microscopy techniques, something like min-flux nanoscopy, which has been quite useful, which means a combination of all of these techniques I've talked about so far. So it's very powerful technique. And also expansion microscopy, which sort of encompasses all of the principles I've talked about nanoscopy at the moment, which basically increases the resolution of these biological samples, believe it or not, by physically expanding the sample itself, which is kind of mind-blowing in itself. And so we can also categorize these techniques under different quantitative methods, where we can understand the spatial organization and molecular interactions, and then stoichiometry of molecules, biological molecules for molecular counting, and also for temporal dynamic studies as well, and so on. Most of my research was based on a single molecular localization microscopy technique, which I've showed before, and there is a specific sub-technique called direct stochastic optical reconstruction microscopy, which is short for DSTORM, which is a handful, like the name itself, but it directly convey the message, how the basic idea of what this technique is about. It basically utilizes the photo switching capabilities of fluorophores to temporarily resolve and reconstruct very high resolution images, which you can see from here. It's a very simple illustration, but it sort of, I think, gives the idea how this technique actually resolve these high resolution images. And so my doctoral research was primarily based on understanding this highly conserved outer membrane protein in bacteria called the beta barrel assembly machinery. Now, it's known to be highly conserved primarily among gram-negative bacteria, but there are homologs among other prokaryotes. And so it consists of these 5 subunits, basically BAM-A to BAM-E. So BAM-A is the core of this complex, and it also been referred to as the catalytic domain. And also the whole complex is being designated to be involved in biogenesis of the outer membrane proteins called beta barrel proteins, which in itself plays numerous vital functions in bacteria, specifically for their survival. So for my research, I used E. coli as a model gram-negative bacterial system to study this complex. And so the elements of the BAM complex activity in the process of outer membrane biogenesis are still understudied. And there's a lot to be known about this. And there are two different propositions and are not mutually exclusive. So in terms of bacterial cell biology, there are two conflicting models concerning the spatially important aspect of the outer membrane protein biogenesis during bacterial cell growth. One model states that the BAM complex, or this complex that actually generate these outer membrane proteins, they're spatially constrained at the midpoint of the cell. And only thereafter do outer membrane proteins move towards the cell poles, as been shown here, and finally accumulate at the cell poles. And the other model basically suggests that the BAM complex could be spatially distributed evenly across the cell membrane and not polarized in particular fashion. But both of these models show that pre-existing material, shown in orange, old material, tend to be forced outwards to the bacterial poles at the end. And the cell division ultimately yield an unequal partitioning of new and old material in their progeny. And so given these differing conclusions of how the BAM complex and the outer membrane proteins reside, we tend to answer some of the, or shed light to some of these questions using our super resolution microscopy technique. So we actually immunolabeled the outer membrane of E. coli, directed these antibodies, which has a fluorophore. They're directed at this complex itself, and we label these cells with these antibodies. And what we saw through the lens of nanoscopy is that the BAM complex is highly organized into clusters all over the cell membrane. And their size and their distribution has been quantified. And we use this term precincts to identify these clusters because we believe is at these points actually the biogenesis of these proteins actually happen. And these are the points actually that makes or propagate the development of the outer membrane itself. So it's quite remarkable what we can see through the lens of nanoscopy itself. And so we also pre-treated these cells with rifampicin, which is an antibiotic which blocks protein transcription. What we saw when we treat these cells with rifampicin is that the BAM complex itself sort of shrink by 20%, which meant that ipso facto, the size of the protein assembly precincts should expand in the presence of active biogenesis of proteins. So this sort of concludes that these precincts are actually actively engaged in protein biogenesis. We also discovered that one crucial subcomponent of the BAM complex known as BAM-B, when we remove this component, the whole clusters or the supercomplex sort of breaks down. So BAM-B subunit seems to be the subunit actually hold these clusters together at the outer membrane. So that was also quite a breakthrough, which we published in 2018. And so this sort of, paved the way to understand these interactions even further using other mutational studies and structural biological studies, and to help identify and support these findings. And we use this photo cross-linking techniques, which basically inserts unnatural amino acids to the genetic code of E. coli, and we are able to identify key residues in this complex that could actually mediate these interactions. So this is actually a very nice way of complementing what we saw in nanoscopy with other biochemical techniques. So what we understand is at the end, nanoscopy helped us to discover these really tiny complexes, which we know to be involved in protein biogenesis. We coined them as assembly precincts, and the observations from these nanoscopy can be complemented with other mutation and structural biological studies. Now, my research is one of many of these research that have been done using these techniques. There's a lot more out there that you can actually read and understand. And so next, I would like to talk about my doctoral research, which also involves the same technique, but looking at a different system. Now, at this point, I'm looking at T cells, which we all know are frontline at protecting ourselves in maintaining homeostasis of our human biology and also in the animal kingdom in general. So T cells are at the front line of maintaining adaptive immunity. So understanding ins and outs of T cell signaling machinery can be quite challenging. And so we actually use these nanoscopic techniques to understand, investigate further into the signaling machinery of T cells. So this is primarily done through understanding the formation of immune synapses, not to be confused with the neurological synapses, but the side of the interaction between an antigen-presenting cell and a T cell creates these type of interactions, which we can readily see, even with a diffraction limited fluorescent microscope. We can see these formations of assemblies of different proteins, which we call as supramolecular assembly clusters. The synapse formation upon the interaction of these two types of cells are dependent on poorly understood signal integration, and therefore the function and the dynamics of all the proteins that are involved are subject to extensive ongoing research. And that is why I'm using single molecule localization microscopy to understand the ins and outs of immune synapse formation. So over the years, in general, fluorescent microscopy was able to more than visualize this spatial organization of receptors in the contact sites of T cells and antigen-presenting cells. In particular, T cell receptors has been shown to multimerize or oligomerize, form these clusters when a T cell recognizes and binds to an antigen, which are derived from obviously pathogens. foreign materials. So these TCR clusters would merge and form these bigger clusters, and with the advent of super resolution microscopy, we are now able to dissect what is within these clusters even further. So in T cells, a lot of these signaling molecules, starting from T cell receptors to a whole lot of other signaling partner proteins, are known to be in this clustered formation. Now, if you look into T cell receptors, generally they're known to exist in these tiny clusters. Upon antigen recognition, they become micro scale clusters. And so, again, with the previous work that been done in our lab, again with the help of nanoscopy, we now know that how we can distinguish different types of clusters. Some of these TCR clusters can be involved in signal propagation and the others would not be. Now, to assess this, we developed different methods, analytical techniques, analysis techniques to identify different densities of these TCR clusters. And we found out that the more denser these TCR clusters become, the more likely they'll be associated with signal transduction. And so there are different biological functions that have been proposed for T-cell receptor clustering. One thing we know that it can actually act as a digital switch, ensuring high fidelity signal transmission across the cell membrane of T cells. It can also increase the affinity towards antigen binding, which collectively known as avidity, which is very important when triggering T cell receptors and triggering a response in a T cell, which could be targeting or eliminating the targeted cell. And also clustering can increase the antigen sensitivity of T cells. This is why an even minute amount of antigens can trigger an immune response. And this is very characteristic to the adaptive immunity. And also there are theories that surrounds the conformational changes in the CD3 complex. The CD3 complex is the signaling domain of the TCR. TCR in itself cannot transmit any signal without the CD3 complex. So there are a couple of conformational changes that happen when TCR binds to MHC, or major histocompatibility complex, which is the antigen presenting molecule. And these conformation changes can be propagated within a cluster. And those are a couple of theories that explain the biological function of T cell receptors. And so what drives TCR clustering? That's also quite a big question in the field. There are different components, such as membrane cholesterol levels, signaling partner protein enrichment in lipid raft domains, and then even interactions within the complex itself has been shown to factor in the level of TCR clustering. And recently we've shown that antigen binding conformation can also affect some degree of TCR clustering. And my question at the moment is how, or if there is a difference in the TCR clustering when T cells express specific TCRs. Now, the conventional T cells express alpha beta TCRs, and we have unconventional T cells as well, which predominantly express gamma delta TCRs. So what we know so far is that a lot of these unconventional T cells have specific functions, and most probably gamma delta T cells that we know are very unique T cell population, and they're rare in secondary lymphoid organs, but they're enriched in peripheral tissues such as skin, intestine, and lungs. And so by rapidly producing large amounts of cytokines, gamma delta T cells make key contributions to immune responses in these tissues. Now, their role in cancer has been quite controversial. You can actually read upon this, but which I'm not going to go through today. Unlike most conventional alpha beta T cells, gamma delta T cells display a large diversity of ligand recognition and docking geometry, which basically the spatial organization when a T cell receptor binds to the MHC complex. And so there are even different types, classes of MHC molecules that we know of, and they present a plethora of antigens ranging from peptides to lipids to bacterial metabolites. And the field itself is still trying to understand a lot of these functions of these different types of MHC presenting molecules. And so lately, I was involved in understanding how an MHC-like molecule called CD1A endo, which is known to be expressed at high density in Langerhans cells and myeloid dendritic cells, which is known to present lipids, including lipid antigens, including phospholipids, glycolipids, lipopeptides, and a whole lot of lipid antigens. We have used this system to understand the ins and outs of the signaling machinery of gamma delta T cells. Now, when CD1A binds to gamma delta TCR, it basically phosphorylates the signaling complex and this T cell signaling machinery. What we've shown here is basically the gamma delta T cell receptors are not that different in the signaling capabilities with their alpha beta counterpart. So at least in this context, we know that there is not much difference between these two types of T cell receptors, and in overall in these two types of T cells in itself. And so, you know, although nanoscopy is a very powerful biological technique, non-invasive, very direct, visual biological technique, there are drawbacks and challenges and even trade-offs. So one thing I need to highlight is that we need to match experimental requirements with the technical performance. Now, diffraction limited microscopy, like normal fluorescent microscopy, has been quite vital in biology and still is. There are so many questions that can be answered using even diffraction limited microscopy techniques like that. Only for specific questions that is required to encompass a higher resolution scale, we can include these type of nanoscopy. If you can achieve these goals without this high resolution, then you simply don't need to use these techniques. And also the complexity and the user friendliness of these systems is actually pretty low. As a user for almost 5 years, 6 years of these techniques, I must say they're not actually user-friendly techniques. So there's a lot of training that goes into it. There are commercialized versions of these techniques are now available, which are more, you know, applicable to a lot of these biological questions, which is quite gratifying. But there are still a lot of challenges even with these commercialized systems. And also with these techniques, one great trade-off is that at this high resolution scale, you tend to get more artifacts. So you need to be able to identify them as artifacts. That's part of the training that you need to go through. And you need to understand that pretty clearly, and not to interpret artifacts as evidence for biological, answering a biological question. And also superresolution imaging should be used in a very non-invasive manner. Now, I sort of talked about nanoscopy being very non-invasive, but that's not entirely true. The different techniques use different, technologies to enhance the visualization. So sometimes it can be non-invasive. So what we need to preserve is the very physiology of these cells, if you need to understand the actual biology within them. And one major concern for me, and I think this needs to be improved in the future, is that nanoscopy is not actually a high throughput screening method, which means you can't really get population level statistics, which means it's very hard to interpret a lot of these evidence that you gathered from nanoscopy, which is, again, kind of a trade-off, but something that would need to be improved in the future if we need to get more higher level of statistics, population-wide statistics, something like flow cytometry does. But again, a lot of these techniques produce enormous amount of data. Within a day, you can gather 20, 30 terabytes of data that needs to be stored in supercomputers. So again, even though with high resolution, there's a lot of problems that comes with that. So with that, I would like to acknowledge a lot of my collaborators, including LeetCode Lab, which I did my PhD at Monash University, Trevor Litgo, my supervisor during my PhD. A lot of these, my doctoral studies is done in collaboration with Monash Microimaging Facility and Bell Lab at Monash Chemistry. My postdoctoral work is primarily based in Gauss Coed Lab at UNSW, and also currently I'm doing a lot of work with Ross Shawn Lab. Basically, Jamie and Ben has been helping me out a lot of in inception of these projects, and also Senthil Armugam at Monash. And so I would like to thank NcodeX for giving me this opportunity to reach a wider audience. Yeah, thank you very much.
Thank you very much, Dilshan. That was very interesting, beautiful, and also very interesting content. And you've managed to get a lot of new findings, and that's pretty impressive, I think. So if anyone's got any questions, any clarifications, post them in comments, and we'll share the questions. Meantime, Dilshan, I would like to ask, so you're using a nanoscopy, and you mentioned you're one of the very few who are using this technology. So it might not be directly related to it, but can you give us a couple of examples? What other researchers who are using this in your lab teams, or not details you're not supposed to disclose, but at least to get us an idea about others who are using this technology right now in your lab?
Yeah, so the technique itself is actually quite old, even though the Nobel Prize was given in year 2014. The inception of this pioneering work was actually done in, I think, 1993. It actually took that long to kind of make these techniques more available and user-friendly for biologists, I guess. What I meant by I've been one of the lucky ones who've been able to use these techniques during my PhD that I know of is that the technique itself, a lot of people use this technique to complement their already ongoing work. What I've done is to start my whole doctoral study from nanoscopy and then dive into other biological techniques. But certainly, right now, people, been spreading the word. And a lot of these nanoscopic techniques have been complementing other biochemical techniques, structural biological techniques, and it's, the applications are still a bit underwhelming, but there are a couple of really good breakthroughs that are made by nanoscopy. In particular in neurology, I think, and I even read articles on cardiology. Scientists are trying to use these type of techniques to understand the molecular architecture of different cells, different membranes. And I think these techniques can be readily viable to be integrated into a lot of other subfields in biology itself in the future.
I think I can agree, because being in neurosciences, I kind of use the similar techniques myself, but it's very useful to actually visualize and bring all these images together. And also, I would like to highlight that point where you mentioned it's not very high throughput. That is something we can actually work on. So
Yes.
we'll get to that in a minute. But before that, so looking into your research outcomes and the new findings, and also looking into the innovation of the world's trending innovations, where do you think your research outputs will go in? Are there any practical applications? So if anyone wants to get into similar research fields, what kind of research questions do you think they can address or they can start looking at?
So, I mean, at least from a perspective of bacteriology, which my doctoral research was based on, I was basically mapping the molecular architecture of outer membrane of gram-negative bacteria. We know that gram-negative bacteria are highly important to understand a lot of these diseases, bacterial infections. So mapping the molecular architecture of outer membrane can probably perhaps give more insights into developing antimicrobial drugs in the future, which may not seem like a direct translational study, but these are fundamental questions, and so to see these answering these fundamental questions and then probably facilitating something like development of antimicrobial drugs may seem a bit far-fetched, but everything stems from fundamental research. And so, yeah, it's, again, as I said before, there are sometimes very specific questions that nanoscopy can address. And sometimes you don't actually need to use nanoscopy. Even normal light microscope can solve some of these questions. You need to understand which type of biological question actually requires that type of advanced molecular imaging. And if it can, then that's a success. Yeah.
That's yeah, so there's huge potential, and technology is advancing, and that means we can address more and more detailed information. We don't know what we'll uncover, and we'll see.
Yeah, I mean, even as we speak, the nomenclature itself within this field, I mean, the abbreviations, the types of nanoscopic techniques that have been developed is quite overwhelming. You know, each day there will be a new article published in Science or Nature saying that, oh, we've reached another diffraction limit with this technique or other technique. So there's constant evolution of these techniques. So my ideal nanoscopy would be to use this at some point in a more user-friendly interface, and then of course use nanoscopy as to a level that we use in flow cytometry, which we know we can get a lot of population level statistics. We're not right there at the moment with nanoscopy, but you know, with the technological advancements, we'll get there pretty soon, I think.
Right. Like, taking out on that, Dilshan, I'm going to ask a question. So you are in this field. So at NcodeX, we are always pushing the interdisciplinary research, and also we're looking at to 10, 20 or 50 years ahead, what research and what products can be developed at that time that will be that would give a really good rate of innovation. So as a user, you always continue to talk about improving the user friendliness and to make it better. So as a scientist, what are there any suggestions you can give to any biomedical engineer or anyone who would develop these techniques so that they can consider during their work, so that they can make your work much more user-friendly and better? What are the points you would tell them?
Yeah, I mean, again, STEM fields, we tend to think they're very integrated in a way. You know, a biologist knows what a chemist does. A chemist would know what a physicist does. But normally it's not the case. A lot of these inventions, and particularly for microscopy, they're made by physicists. So their idea of understanding cellular system maybe comes from fascination, but the actual users of that technique would be the biologists. So there is a gap in terms of understanding the technicality of the technique you use, and what type of biological question we try to answer as a biologist. So the technical aspect is something that I feel like biologists would be blinded to. But it's sort of in the nature of a lot of biologists. We just do not tend to, or even try to understand the ins and outs of the mechanisms that in play of the biological techniques we use. I mean, even If you look into a medical perspective, a medical professional would not know the ins and outs of an MRI machine, but they know how to use it. But at some point, I think a physicist and a biologist or even a chemist should have that conversation together, integrate themselves, and in that way, I think, and the emerging fields of biophysicists, a biological application in physics, it kind of speaks volumes into this cross-disciplinary work. So again, I think maybe I'm trying to answer this question in a little bit of like a vague manner, but what I'm trying to say is that scientific research doesn't go to fruition with just one field. You need to have interdependence within other fields, especially in STEM fields. In that way, the more multidisciplinary research becomes, the more applicable that would be, and more translational in that sense. So maybe the physicist doesn't know what the biologist does and vice versa, but there has to be some kind of connection. It has to be multidisciplinary in a way so that they can both achieve the same goal.
So they do need to get into one round table, have a quick talk.
Absolutely. That's the way to go.
Well, things need to change. That's perfect. So while we're on this slide, I see you've been working with a lot of different people, a lot of different lab groups and research teams. So if is there any advice you would give to any young researchers, especially on networking, how this networking has helped you achieving your research goals and career goals?
Yeah, so I think as a lonely graduate coming from Sri Lanka, I mean, it's not easy to come into a foreign land and start integrating yourself. I'm talking about life in general, and then step ahead with research. As we all know, scientific research, that's a totally different language. You need to have the discipline and some form of learned experience to have that kind of conversations with scientists and colleagues. But the bottom line is we're all human. You know, just be truthful, respectful to each other, and just, we're all trying to learn. And I think as a scientist, we need to be very humble in that sense that we are still learning. We don't know anything for sure. These are just observation-derived conclusions. I don't even say them as conclusions because they tend to interpret in various ways with new knowledge. And so networking with people, it's not easy, but you learn it. But you need to be respectful for each other, and especially, be true to the science and what you're trying to achieve. And that way, I think you can go a long way. And these ties you make, and particularly the ties I made during my PhD, they still last today. So that's something I do cherish. And it's basically participating in conferences, outreach programs, not confined to laboratory space, which a lot of PhD students try to do this, but going outside, networking with your own peers is actually very beneficial. And so, yeah, I think I hopefully answered that question.
Yeah, absolutely. So it's like you said, we may enter it alone, but we don't have to stay alone. There are other people as well. We can obviously get in touch and we can help each other out, not just that they help us out at the same time we are being there for them as well. And one final thing before we wrap up. So at the moment, Sri Lanka is facing a little bit difficult time. And just today morning, we had a session workshop for them to help out with any of the, to develop their careers. And are there any general advice you would give to any young researchers who wants to get into scientific career, who's doing maybe A-levels or undergrads? And what would be your one final thing to one message to them?
I think as a society, I mean, in Sri Lanka and as a whole around the world, there's this notion that science is, always produce things positive results, and it's very edgy, and everything comes to fruition very easily. But that's not the case. And especially in STEM fields, it's not easy. There's a lot of analytical thinking that involved in it, and it's something that some people learn over time, and some people may take a long time to learn this type of analytical thinking. But as a society, I think in Sri Lanka, we were bred to kind of think in these terms. Getting to STEM fields is quite gratifying, especially for our parents, at least. But when you get into science, and if you do have the guts to make it into science, there are going to be a lot of ups and downs. It's not going to be, distinctions and honors every time. It is a tough business. Doing science is not easy, but never giving up is something I think that's the only message I think I can give anyone. There is no standard for intellect, I think, if you want to get into science. You just need to have the curiosity. And if you have that, I think you can go a long way, because a lot of scientists I've seen kind of lose it over the course of their life. But something like the discovery of penicillin, that was an accidental discovery, and so an accident that made a human life a million times better than we had, pre-antibiotic era or something. So those type of history, I think people need to, and especially young graduates or A-level, O-level students need to read upon these things and get some kind of inspiration that STEM fields are actually the foundation in a lot of these technological advancements. I mean, we're talking in a not even a post-pandemic era. We're still in the pandemic. We're not cleared out of it yet. But the rapid technological advancement that was made in vaccine production in itself, I think like within under 6 months or something, that's quite inspirational to the whole world, that science is producing and saving lives, to be honest. And particular fundamental research, I think, need to have more funding in that way. and so that these would be more translational into, front line of medical discoveries at some point. And so, yeah, never give up. And if you want to do STEM, get into STEM fields. Never give up. And I think basically it's gonna fulfill your life in a way. Yeah.
I think everyone can agree on that as well, because yes, there are ups and downs, but at the end, that thrill of discovering something that is not comparable to anything, right? So that's that contribution you can make. You never know, you might discover something, you might change the world. So get into science, and you'll grow a thicker skin as you go along, as you face these problems.
Yeah.
Right?
Absolutely.
This has been very fruitful, Dilshan. Thank you very much for your time. And thank you very much for everyone joining with us. And if you have any questions, if you want to get in touch with Dilshan, by all means, reach out to NcodeX, and we are happy to get you connected to him. And meantime, I think most of this work has already been published, and the work is shared in the description. There are links to the publications. Go ahead and have a look. And if you want any more support, we're happy to help you out as well through NcodeX. And that concludes our 13th episode, and we will join with the 14th episode of PhD Talks by NcodeX in October 2022. And for now, have a great rest of the day. Have a great rest of the week, and we'll see you soon. Thank you.
Thank you very much.